A pleasant morning to everyone, to my thesis advisor, Dr. Lalaine Grace M. Maghanoy, to my panel committee, Dr. Merced G. Melenchon and Dr. Mark Lloyd G. Dapar, to my classmates and guests, good day. I am Jiraldine V. Novecio from BS Biology 4A, specializing in microbiology, and for today, I will present my undergraduate thesis proposal entitled In Vitro Cytotoxic Potential, Antimicrobial Activity, and Phytochemical Screening of Medicinal Melanolipis Multiglandulosa Crude Leaf Extract. This would be the content outline of my presentation. First is the introduction, then the objectives of the study, methodology, and references. I will begin with the introduction. So herbal medicines have been the basis of treatment and cure for various diseases and physiological conditions in ethnomedicinal practices. Currently, medicinal plants gaining a lot of importance because of efficacy they have been showing in the traditional healing. Ethnomedicines are in demand as they are inexpensive and have much less side effects. The World Health Organization has accounted about 60% of the world's population relies on herbal medicine, and about 80% of the population in developing countries rely almost on it for their primary healthcare needs. One of the plant whose parts have been widely used for medicinal purposes, both locally and across the continent, is the leaves of Melanolipis multiglandulosa. It is a tree or shrub belonging to the Euphorbiaceae family. It is commonly called a womb or alim plant with height approximately 4 to 20 meters. It is native to southern Japan, Taiwan, Thailand, Malaysia, Indonesia, and Philippines. According to Apostol et al. 2016, Melanolipis multiglandulosa leaves are traditionally used to treat some diseases such as poultice, against different kinds of scurf, as an anthelmintic against parasitic worms, as an anti-cough decoction, as a drink to treat constipation and tuberculosis, and mixed with some bark and flowers as temporary relief for chest pain and fever. The basis of my study is from the article of the Par et al. In 2020. It was stated that the Melanolipis multiglandulosa is one of the plants used by Manobo tribe for medicinal purposes. And according to the study reported by Lai et al. In 1996, the methanolic extracts of the roots of Melanolipis multiglandulosa contain fridolin, oleanolic acid, beta-amycin acetate, campesterol, stigmasterol, beta-sitosterol, phytosterol glycosides, and D-sucrose. And recently, the study of Apostol et al. 2016 reported the isolation of taraxyryl, fatty acid ester, squalene, 3-alkanoic acid, carotene, a mixture of beta-sitosterol and stigmasterol, long-chain fatty alcohols, and long-chain hydrocarbons from the leaves, and triaglycerols and long-chain saturated fatty acid esters from the twigs of Melanolipis multiglandulosa. However, most herbal medicines have only limited scientific evidence to support their safety and efficacy as stated by Bent 2008. There is no scientific basis or evidence of the antimicrobial, cytotoxicity, and phytochemical constituents of the leaf extract of Melanolipis multiglandulosa. Thus, this study is to validate the claimed antimicrobial property, cytotoxic potential, and phytochemical constituents of Melanolipis multiglandulosa leaves. This study hypothesized that crude leaf extracts of Melanolipis multiglandulosa may possess antimicrobial activity against selected pathogens, no toxic effect on erythrocytes from human blood, and may contain phytochemicals that can be used for drug discovery in the future. The objectives of the study generally aim to determine the cytotoxic potential, antimicrobial activity, and phytochemical constituents of the leaf extracts of Melanolipis multiglandulosa. Specifically, it aims to collect the leaves of Melanolipis multiglandulosa at Poblacion, Valencia City, Bukidnon, extract the powdered leaves of Melanolipis multiglandulosa using methanolic and ethanolic solvents, to evaluate the cytotoxic potential of the leaf extracts against erythrocytes collected from human blood, to assess the antimicrobial property of Melanolipis multiglandulosa crude leaf extracts against selected bacterial and fungal strains by agar-Dis diffusion method, and to determine the presence of alkaloids, anthraquinones, flavonoids, saponins, and tannins from the leaf extracts of Melanolipis multiglandulosa. This study will help to provide scientific basis on the efficacy and medicinal claim of Melanolipis multiglandulosa leaf extract, and can help to verify if its leaf extract can be potential antimicrobial, cytotoxicity, and contains phytochemicals. Moreover, the information gathered in this study can be useful in pharmaceutical industry for drug discovery and can be used for further research as a reference. Furthermore, this study will help to contribute to the society and for additional information on the benefits of consumption of Melanolipis multiglandulosa for health purposes. The study will be carried out using the leaves of Melanolipis multiglandulosa, since leaves are more abundant than other parts of the plant. Other extraction procedures will not be utilized aside from methanolic and ethanolic process because they are not harmful to the body. The hemolysis assay will use red blood cells or erythrocytes, since it is the most abundant cells in the human body, and other cytotoxic assays will not be included. The Dis diffusion method will be performed for antimicrobial assay, since it is inexpensive and has the ability to test enormous numbers of microorganisms. For the phytochemical test, it will be done qualitatively, and since this method is expensive, the test will be limited to detect only the alkaloids, anthraquinones, flavonoids, saponins, tannins, and furthermore, other possible therapeutic effects of the plant will not be studied. This is the schematic diagram of methodology. First, the leaves of Melanolipis multiglandulosa will be collected at Valencia City, Bukidnon, and will be identified at the Museum of Central Mindanao University with the help of experts. Then, the collected leaves will be prepared for extraction. Next, the solvent extraction will be carried out after the leaves are powderized by using the methanol and ethanol solvents. Then, the extracted sample will use for cytotoxic assays, and it will be done by hemolytic assay using the human red blood cells or erythrocytes to assess whether the plant is safe or toxic. Then, if it has no toxic effect of the red blood cell, it will then further examine on the Dis diffusion assay for antimicrobial and antifungal activity of this plant. And lastly, the extracted sample will be tested for the detection of phytochemical constituents such as alkaloids, anthraquinones, flavonoids, saponins, and tannins. So this figure shows my schedule to conduct my thesis proposal. This Gantt chart will help me to manage my time and to be able to finish it on time. And this slide shows the references of the study. And I guess that ends my presentation. Thank you for listening.